This lesson covers structures and processes that happen at the cell level. We will be covering cell structure, mitosis, meiosis, and the introduction of certain horticultural terms. Plant cells contain a number of different structures, including a cell wall, cell membrane, plastids, vacuoles, and a nucleus. The cell wall gives the plant structure so that it can stand upright. Animal cells do not have cell walls, so they would not be able to move. A cell membrane is a gatekeeper. It allows things in and out. In some cases, it can pull things in and expel others. Plastids are organelles that produce carbohydrates such as sugars and starches. There are different types of plastids. For example, chromoplasts produce pigments, and chloroplasts contain chlorophyll for photosynthesis. Animal cells do not contain plastids. A vacuole is a membrane-bound sac. In plant cells, one of its roles is to produce turgor pressure in the plant. If the plant is well watered, the vacuole enlarges and produces rigidity in the plant. If the plant is under moisture stress, the vacuole is reduced in size and the plant wilts. Though animal cells have vacuoles, they are much smaller and play a different role. The nucleus is a structure that contains the genetic material of the plant. Think of the genetic material as a blueprint. It tells the plant how to design and build another cell. The nucleus looks like a little sphere within the cell and contains the genetic blueprint of the plant. Every cell in a plant has the same blueprint. It doesn't matter if it is a leaf cell or root cell or any other type of cell. The blueprint remains the same. The difference between a stem cell and root cell is not in the blueprint, but in what part of the blueprint is turned on and used. Think of using a blueprint when building a house. When building the kitchen, you may have the blueprint for the entire house, but only the portion of the blueprint that describes how to build the kitchen is used. The fact that the entire blueprint is present in every type of cell but one means that we can easily make exact copies of plants. We do this by using various types of what we call vegetative propagation. These can include grafting, budding, cuttings, layering, and air layering. We will look at vegetative propagation in a bit more detail later. First, we will cover cell division starting in the next slide. When a cell divides, a copy of the complete blueprint is made, and that copy goes into the new cell. Actually, cells contain two complete blueprints, one from the father and one from the mother. These blueprints work together to determine what characteristics a plant has. The fact that there are two blueprints becomes very important when we talk about producing seed, which we will cover later. Let's illustrate this with examples from the genetics of people. The first example we will use is for eye color. Let's assume the blueprint from the father says the eyes of the child will be brown, and the blueprint from the mother says the eyes will be blue. What will be the color of the eyes of the child? The child's eyes will be brown. This is an example of dominant and recessive. Brown eye color is dominant and blue is recessive. So if the father's blueprint says the eyes will be brown and the mother's blueprint says the eyes will be blue, the eyes will be brown. In such cases, one blueprint overshadows the other. This does not always happen like this. For example, a child's height may not match either the father or mother, but could be anything along a continuum. Why is it important for cell division to take place? There are actually a number of reasons. First of all, if cells can't divide, a plant can't grow. There's a limit to how large an individual cell can become and still perform its purpose, and so cells can't just grow larger, but must divide. Many pre-emergence herbicides make use of this characteristic by inhibiting cell division. If a seed germinates, but the root cells can't divide, the root system doesn't develop, and a very young plant dies from lack of water uptake. Just remember that mitosis produces daughter cells that contain the exact same blueprint that was in the mother cell. The cells may look different and do different things. For example, a leaf cell does something different than a root cell, but the blueprint is exactly the same in each. This is why we can make exact copies of plant through using what we call vegetative propagation. Plants are propagated vegetatively through such procedures as grafting, cuttings, budding, air layering, etc. When you use these methods, you're making an exact copy of the original plant. Sometimes people will ask how to get an apple tree like they found in Grandpa's orchard. No one remembers the name of the variety, so they can't just buy another one. In such cases, seed will not work. Though you will get an apple tree from the seed, it will not be an exact copy of the parent. If you want an exact copy of the apple tree, you must vegetatively propagate it through a process such as grafting. Another example why vegetative propagation is important is in disease resistance. Most American elms are susceptible to Dutch elm disease, which has decimated American elm populations across the U.S. However, there have been a very few American elms that were naturally resistant. Those have been identified and propagated by cuttings. Since the trees grown from cuttings are genetically identical to the parent, the progeny also have Dutch elm disease resistance. The varieties listed here are all true American elms, not hybrids, and all have excellent Dutch elm disease resistance. Princeton is the nicest looking tree when young, but Prairie Expedition is a bit stronger wooded and is less likely to be damaged in wind and ice storms. Another example of the importance of vegetative propagation is in the production of cottonless cottonwoods. Cottonwood trees are dioecious. In other words, there are male and female cottonwood trees. Not all trees are dioecious, but a few, including cottonwood, are. Since the cotton from cottonwood is only produced by female trees, planting a male cottonwood can avoid this problem. So how do you tell a male cottonwood from a female cottonwood when they are 3 feet tall? That is a trick question because you can't. You must wait until the tree produces flowers. A tree that produces male flowers is then propagated via cuttings. Since all the progeny are male, these trees can be sold as cottonless cottonwoods. Look at these two squash. The one on your right is an acorn squash. The one on the left looks like it crossed with something. So what happened? People often assume that a strange squash appeared because one of the squash planted in the spring crossed with something else, and this is what was produced. That is not what happened. Remember, every cell of the plant, including every cell in the fruit, has exactly the same blueprint that originally came from the seed. So what happened? There are 3 possibilities. First, this plant was a volunteer from squash planted the previous year. Or the seed you received from the seed company was bad. Or the third possibility was that this was a mutation. Mutations are rare, so it's likely one of the other 2 possibilities. Understanding mitosis can help master gardeners understand why certain things happen the way that they do. Meiosis is another form of cell division that is used to produce pollen and eggs. Meiosis produces cells that are the exception to the rule. They do not produce cells with exactly the same blueprint. Each pollen grain and each egg has a unique blueprint. Why is this so? During the process of meiosis, the male and female blueprints trade information. This happens at random locations so that each pollen grain and each egg ends up with unique blueprints. Also, each pollen and egg has only one blueprint, so that when they join together during fertilization, they'll return to the normal 2 blueprints, one from the father and one from the mother. Why is meiosis important? Without meiosis, plants couldn't produce seed, and seed allows plants to adapt. Remember, each seed from a plant differs genetically from every other seed produced by that plant. For example, one seed might confer a bit more drought tolerance, and another additional cold hardiness. The Kansas Forest Service sells trees for windbreaks in Kansas. One of the species they sell is the Austrian pine. They are careful as to where the seed they buy originates. Usually they buy seed that comes out of Missouri and avoid places like Georgia. Though both sources of seed are for the same species, they differ in how adapted they are to Kansas conditions. Plants that are better adapted to local conditions produce more seeds than those that aren't. Over time, a biotype is produced that is well adapted to local conditions. Therefore, Austrian pines from Missouri are more likely to survive than those from Georgia. Even if plants can't produce seed, for example a male Osage orange tree, it can still be propagated vegetatively through cuttings, grafting, budding, or various other means. However, vegetative propagation results in exact copies of the parent with no variation. Meiosis produces daughter cells that have 1/2 as much DNA as the parent. The egg or pollen grain only has one blueprint. When the egg and pollen join to form a new plant, we return to the normal 2 blueprints, one from the male and one from the female. Here's what is important regarding meiosis. Seeds from the same plant are not identical in their blueprint because the genetic material was all jumbled in meiosis. If you want a plant just like the parent, you do not collect seed and plant it, but must use vegetative propagation such as cuttings, grafting, budding, layering, or some other vegetative technique. There are a number of common terms you'll hear as a master gardener, and knowing the definitions can help you accurately communicate what you wish. We will start with the definition of a variety. A variety is a selection within a species that comes true from seed. It is different from a cultivar, which we will discuss later. For example, thornless honey locust and thornless Osage orange will usually produce progeny that are thornless. Other names for Osage orange are hedge trees and bodark. Osage orange is an interesting tree. Its native range is actually quite small, includes a small area in western Arkansas, southern Oklahoma, and portions of East Texas. However, early explorers such as Marquette and Joliet found Osage orange trees growing near Osage Indian villages. The wood from Osage orange was highly prized for making bows. The bow blanks and finished bows were used as items of barter between the Osage tribe and various other tribes. An early report said a well-balanced bow was worth a horse and a blanket. Osage orange bows were used by the Shawnee and Wyandot tribes in Ohio and the Blackfoot tribe in Montana, and so is traded over a very large area. Osage orange was also a valuable tree for settlers as it was used for fence posts as well as making hedgerows. Osage orange trees are so thick and thorny that cattle won't go through them, and so a line of trees can become a living fence. There are varieties of Osage orange available as landscape trees that are male, so they don't produce fruit, and also are thornless. Examples include Wichita and White Shield. If you want a tough tree that isn't bothered by insects or disease, Osage orange would be a good choice. A cultivar is a shortened term for a cultivated variety. Cultivars do not come true from seed. For example, Crimson Sweet watermelon was developed by Dr. Charlie Hall while he was at Kansas State University. If you plant seed from Crimson Sweet, it will grow and produce a watermelon, but the fruit will not be as good as the parent. All hybrids are cultivars, as none will come true from seed. Another term that actually is not so common is tissue culture. It is a procedure where we grow new plants from the very tip of a stem. What you see in the photo is a plant growing in a test tube. The material in the bottom is agar, a jello-like material that has all the nutrients needed to support plant growth. You can see a number of new plants have started from the original plant material. These tiny plants can be removed, split into individual plants with each plant placed in a new test tube with agar. Each of these plants will also produce a number, so new plants can also be divided. In this way, you can start with one plant and end up with thousands if you wish. Once you have the number of plants desired, each plant can be potted up and sold. Obviously, this is a complicated and expensive process. Why would you use it? Let's look at 2 reasons. The first is to clean up diseased plant. Strawberries are notorious for carrying diseases which don't kill the plants, but cut down on vigor and reduce how many fruit they produce. With tissue culture, we can take a diseased plant and produce healthy progeny. How can that be? Shouldn't a diseased plant produce diseased progeny? Usually that would be true, but isn't in this case. Remember, we're taking the very tip of the stem, which is all new growth. Disease hasn't had enough time to move into the tip, and so the tip is clean. If we keep everything sterile, we can produce as many disease-free plants as we wish. Another way tissue culture is used is to produce progeny identical to the parent. We mentioned a number of means of propagation that produce progeny identical to the parent, including grafting, budding, cuttings, etc. However, there are some plants, such as orchids, that none of these methods work well. We can use tissue culture to produce progeny that are identical to the parent at a relatively cheap price. When I was a kid, an orchid was very expensive because they were so difficult to propagate. Now we can find them at grocery stores at a very attractive price. This is due to the ability to produce numerous plants inexpensively via tissue culture. I've raised blueberries for over 25 years. When I started, I want to make sure that it didn't bring in any diseases. No one near where I lived raised blueberries, and if I brought in clean plants, I shouldn't have any blueberry-specific diseases. The blueberry on your left was the size of plants I received in the spring. The one on the right is the size these small plants grew into by fall of the same year. Starting with clean plants allows exceptional growth. We said earlier that plants do not come true from seed. In other words, a plant grown from seed will not have the same characteristic as the parent. An inbred line is the exception to this rule. An inbred line is produced if a plant is crossed with itself a number of times. Variability is lost, and the progeny begin to share the same characteristics as the parent. This is because the blueprints from the father and mother become nearly identical so that the jumbling of information in meiosis doesn't hurt. Heirloom plants such as Cherokee purple tomato are inbred lines. Most inbred lines must be isolated from other varieties of the same species so that the plants must cross with neighboring plants of the same variety. However, tomatoes are an exception and can be planted with other varieties of tomatoes without consequence except for potato-leaved or currant tomatoes, which must be isolated from other potato-leaved or currant tomatoes. A hybrid is the result of two inbred lines being crossed. So why would you want to do this? Well, hybrids are larger, more robust, and more productive than either of the parents. The increase in the amount of fruit produced is enough to more than make up for the extra cost of seed. However, you should not save seed from a hybrid. Plants will grow, but will not share the same characteristics as the parent. This lesson is one of the most difficult of all that we will cover in Master Gardener training. Don't worry if this is confusing. As you grow as a horticulturist, things will begin to fall into place.